They're actually operated by a person, just literally, it's kind of like a post with a weight on it, and you push or pull this weight up and drop it, and then it sort of hammers a tube into the ground. And you can get down to roughly a meter, it's like 3 feet, pretty easily. If your soil is fairly loose, you can get deeper than that, but turns out the soil in these areas is pretty well cemented. It's actually got a loose layer on the top, and then it's pretty well cemented below 1 meter. So we focused on 1 meter cores. So once we then got those samples, we did various things with them. This is an instrument called the Terra X-ray diffraction spectrometer. It's a field portable X-ray diffraction spectrometer. X-ray diffraction is the standard laboratory method for determining mineralogy of samples. And the Terra is a commercial version of an instrument that's going to be flown on the Mars Science Laboratory. So this is actually a photo op type shot of using the Terra in the field in a spacesuit, and the instrument is easy enough to operate that you can actually do that. But in practice, we used it in the laboratory. And then the other thing that we did was we actually did biological extractions and extracted DNA out of the samples in the laboratory, and this is Susanna Dorado, part of my crew, who was the biologist doing that work. Okay, so, and then in addition, the soil cores were brought back to the laboratory and sent, back to Earth, and some additional work was done. This shows an organic analysis from each one of these units. And I don't really expect you to understand what's going on here, but there are some surprises. The Mancos Shale is quite not unexpectedly very organic rich compared to the other soils. And in fact, none of these are organic rich compared to your garden soils. They're only all less than 1% of organics. But compared to the other samples, the Mancos Shale is fairly organic rich. The Morrison Formation, this is the one that the HAB is sitting within, there was no organic signature detected. And this was a surprise to me because it's actually more organic pore than, for example, the Dakota sandstone, which is a porous sandstone, and because the Morrison Formation is filled with fossils. It is literally, there are dinosaur bones visible from the habitat. So it's very surprising to me that there's literally no organics in this material. So we're learning new things by getting out there and taking samples and actually doing things with them. This is an example of the biological sampling. Again, the 5 different geologic units we looked at, and the different distributions of bacteria in the different units. Again, there's some surprises here. Units number 1 and number 4 were both from the Mancos Shale, and you'd expect, or I would have expected, that the distribution of the different types of biology in those 2 Mancos Shale samples would look more similar compared to other types of units. And yet, I've got 2 samples that are almost identical in terms of their distribution of biology, and one of them is from the Somerville Formation and the other is a Mancos Shale formation, but neither one of them looks at all similar to the other Mancos Shale. And these things are roughly 100 million years apart in geologic age. So I don't really understand what's going on here, but the point is that it's usually when you have a puzzle that that you start learning new things. Okay, so so that was what we did kind of initially was collected the soil cores, and after we had done an exploration of the regional area, we picked some sites that we thought might be interesting to drill. We then did a geophysical survey of one of those sites. This is using an instrument called the CRUX Experimental Ground Penetrating Radar System. Here it is being towed by a person wearing a spacesuit, and here's a close-up picture of the sled carrying the ground penetrating radar. The system is really just a couple of dipole antennas with some electronics to read the signature. So here's the feature that we're actually interested in studying. So this is really it's a Google Earth view, but it's effectively the aerial photograph looking at this structure. The hab is over here somewhere, and you have this kind of arcing positive relief feature. It sticks up above the surroundings, and it arcs in here, and then it meets this cliff wall. So here's the hab, and immediately behind the hab is this basically nearly vertical wall. It's not quite vertical. You actually can climb up it, but it's a very steep slope up to a bench table top. So this arcing structure is intersecting that. And this is actually sitting at the intersection and looking down at the arcing structure. So this is marked on maps that crews have constructed at the habitat as the Kissing Camel Range. I actually thought it was more like a snake. The hypothesis is that that feature is an inverted channel. And there are inverted channel deposits that have been discovered on Mars that look very similar. So what we did with this ground penetrating radar is we did a traverse across the intersection where that arcing structure meets the hab ridge. So here's the actual collection. Again, the couple of dipole antennas and the readout electronics. And here's where we did the traverses, and again, here's the structure. And so the idea is that if we were correct, we would expect that this would extend back underground, under this ridge line. And here's the GPR data. And sure enough, as you traverse across, let me just back up here. So basically we did a traverse this direction, and then we turned around and traversed back the other way. And that's what these two plots are, and I've flipped one of them so that they line up in the same direction. And what you see is that you see these structures, and you see them in the same place on both the traces, and this trace is a little noisier than that one, but you're seeing the structures underground in the same places, and you're seeing them pretty much at the intersections where you would be crossing the feature. So again, it seems like this is a pretty good indication that in fact this structure does extend back underground. Another thing that is a very interesting Mars analog and that was we discovered at this site was that the not just at this site, but at many locations in the area of the hab, we found these little round concretions, and they tend to happen where these buried or inverted channel deposits are located, and we've actually now mapped them. The reason that these are interesting is that concretions like this were found at Sinus Meridiani on Mars, and in fact this is probably, to my knowledge, the best analog anywhere on Earth of concretions like the Sinus Meridiani deposits. They're widespread at the hab region, and they seem to be associated with the inverted channels. Here's a map showing the various places that we sampled these, and again, this is more or less the ridge line, and these channels or channel deposits basically were flowing east-west. This is actually north-south here. So their regional extent and they're outcropping in many places, and they are not geochemically similar, but they're morphologically similar to the deposits at Sinus Meridiani on Mars. Okay, so moving on to another one of our activities, which was testing this instrument called the Mars Underground Mole. This is analogous to the push core, only it's a robotic system. So basically, this is a system that self-hammers, small robotic system, and it hammers its way underground. So we took this out. Once we, again, had figured out what the different geologic units were, we deployed this at 3 of them. This is a system that weighs 2 kilograms. Well, actually, the mole itself weighs 2 kilograms. The system mass is more like 15. It operates on 2 watts of power, and it carries light collection optics for a Raymond Spectrometer. That's a type of mineralogy instrument. So here we are in the Jurassic Somerville Formation operating it. And here's another place that we took it actually up onto the debris slope of one of these Manco Shale cliffs, and set it up right up here, way up on the debris slope. The reason we did that was that this was the only place where the soil was loose enough to be able to get the mole to penetrate to its full depth, given the amount of force that it has to pound its way into the ground. And this is what the data looks like. It's actually recording its depth as a function of time. So you can see different things happening, and in principle, if there were layers in the subsurface, you would see differences in slope, which would basically correspond to rates of penetration. As it turns out, this stuff was quite uniform, so the slope doesn't really break. But in any case, we were able to get it to penetrate 65 centimeters. Its total length is 50 centimeters, so we were able to bury it in the material there. And also it has a little door that opens once it's buried in the subsurface, and collects samples that way, and we were collecting samples with it also at these sites. Here's a picture of somebody holding it up in the field, and that's actually the Raymond Spectrometer element of it. Okay. The other thing we did is we did some tests with a human-operated coring drill, and this actually was something we bought commercially and just tried it out at MDRS. I was quite surprised to learn that the human-operated drill system was flexible and fast and easy to operate, even in the simulated spacesuits. And I think personally that this is the right way to proceed in human exploration. Here's some of the places we drilled. Several different places in the Morrison Formation, and one of the places in the Somerville Formation. Primarily what we were interested in at that point was getting samples of the different geologic units and in studying the concretions. So in fact, one of the things that I became curious about was wherever you see these concretions, usually you just see them in a pile on the ground, but they actually are in outcrops. I mean, they're embedded in rock. And the question is, are they actually forming on the surface of the rock, or do they, are they, I guess I would call it well mixed. Are they, do they go down through the center, and can you find them? Because when you see them on the surface, they're actually a different color, so they stick out, and it's easy to spot them. So we did some drill cores through a concretion-bearing